you. Welcome to all of you in our session 2, day 2 lecture series. And we have 2 lectures in this session. Our first speaker is Dr. Gautam Pramanik. He is currently working at UGC DAE Consortium for Scientific Research, Kolkata Center. And he will be talking about synthesis, characterization, and applications of near-infrared emitting nanoparticles. And let us first introduce him. Dr. Gautam Pramanik got his MSc in chemistry from Indian Institute of Technology, Madras in the year 2008. And then he worked for 2 more years at Dr. Reddy's pharmaceutical company, Hyderabad. And he completed his PhD from Germany and at the university, ULM University. And his postdoc is from Prague, Czech Republic, and the institute name is Institute of Organic Chemistry and Biochemistry, where he was my colleague also. And his current research topic includes synthesis, characterization, and understanding of the properties of nanoparticles for various applications. With this little introduction, I would now like to hand over to Dr. Gautam Pramanik for his talk.
Okay, thank you, Professor Devotri, for the introduction. So, can I switch on my video?
Yes, sir. No problem.
Yeah. Good afternoon to everybody. So myself, Dr. Gautam Pramanik, I'm working as a scientist D at UGC DAE Consortium for Scientific Research, Kolkata Center. So today I'm going to talk about synthesis, characterization, and application of near-infrared emitting nanoparticles. So the outline of my talk is the following. So in the beginning, I will talk about advantages of near-infrared nanoparticles in bioimaging. Then I will talk about 2 types of nanoparticle. One is near-infrared emitting gold nanocluster. Another is near-infrared emitting color centers in diamond crystals. So the gold nanocluster, it shows a size-dependent optical properties. And whereas the color centers in nanodiamond, it shows the fluorescence due to the defects in the diamond crystal. So in the gold nanocluster part, I will talk about the size dependence of the optical properties of gold nanoparticles and nanocluster. Then I will talk about surface mediated ligand tuning of optical properties of AUNCs and application of photoluminescent AUNCs in bioimaging. Then I will show you how with the change of subcellular localization of gold nanocluster is observed by design. And in diamond nanocluster part, so I will introduce you to different kinds of color centers in diamond, and then I will show you how we create these color centers in diamond, and then surface functionalization of nanodiamond and the application of fluorescent nanodiamond for sensing. And at the end, I will like to talk briefly about my institute because this might help you in your research also. So let me begin. Advantages of near-infrared nanoparticles in bioimaging. So as you can see, there are many fluorophores inside our body, inside the cell, which absorbs in the UV and the visible region. And in the NIR region, so which is 650 to 1350 nanometer, which is called NIR region, this absorption is quite low. So if you have a fluorophore or fluorescent molecule which emit in this region, so your absorption will not be absorbed by the intrinsic fluorophore. So you can see a better signal to noise ratio. As you can see here, this is a mice which has been cutted, and you can see when you excite with the blue laser and put a green filter, you can see there are a lot of fluorescence coming from this mice, which is the intrinsic fluorophore. And when we put a red filter and excite with green laser, then you can see the fluorescence decreases, but still there are some fluorescence left. But once you put the NIR filter, you can see there is no fluorescence left, completely black background. So now if you have a fluorophore which is emitting in the NIR region, so you will get much better signal and you can see the biological events much better. So that's why, you know, last 2 decades, there has been huge research has been done in this area. This is a multidisciplinary area. So as a chemist, we contribute in synthesizing different NIR emitting nanoparticles. But this is this whole bioimaging involves physicists, biologists, chemists, engineers. So it's a multidisciplinary field. So, and you can see the near-infrared nanoparticle, the fluorescence, it can travel to the much deeper into the tissue. As the wavelength, we go to higher wavelength, due to the lesser scattering and low absorbance, the light, you can get light from much deeper tissue. So this is very attractive for in vivo mice imaging. So there are many near-infrared dyes which are known, like cyanin-based dyes, methylene blue, squalene dyes, or BODP dyes, or rhodamine, which has a silicon substituents here, silicon rhodamine. So and among them, even this indocyanin green, it is actually US FDA approved. It used for different surgical application or ophthalmic treatment. But however, these dyes are, it photo bleaches readily. If you shine a laser on it, it photo bleaches after a few minutes. So you cannot study these dyes for longer period of time. And these dyes have a very low quantum yield. And also, as you can see, these dyes has a quite a high amount of benzene rings, so which makes them very poorly soluble in water. So, and their quantum yield is also quite low, and these dyes are rapidly cleared by our renal system of the body. So you cannot also study for a longer period of time. So, last 2 decades, there has been extensive research has been done on nanoparticle which emitting in the near infrared region. It has several advantages, like these are photostable, and this has a long luminescence lifetime, and we can tune the absorption emission also, and it has a large surface area, so we can attach many targeting ligand, drug molecules, DNA, etc. And this the biodistribution of these nanoparticle in the living animals, say in mice, can be tuned according to the size, shape, charge, so which helps for targeting. Here are a few examples of NIR photoluminescence nanoparticles. It can be, organic fluorophores, as I showed you in the last slide, and it can be fluorescent protein, like, the GFP, YFP, which fluorescence in green and yellow region, but there are recently, there are some fluorescent protein has been discovered which emit in the near infrared regions, such as M. Neptune, tag RFP 657. And also there are polymeric nanoparticle, such as polyfluorine or poly-p-vinylene and fluorescent bead. So there are a lot of fluorescent bead where the fluorescent dyes has been coated with a fluorescent polymer, and so on. So there are also kind of metal-based semiconductor like cadmium telluride, lead sulfide, cadmium selenide with zinc sulfide coat. Another one is copper indium selenide. So this is also one which we are working on in our lab. Silicon nanoparticles, and also the defect centers like NV centers and silicon centers in diamond, as well as carbon-based nanoparticles such as single-wall carbon nanotube, which emits in the near-infrared 2 region like 1240 or 23 some. And also the metal nanocluster like gold, silver, copper, which is very few atom like 15, 20 or 25 atom, which gives fluorescence in the near infrared region. There are other rare earth metal based nanoparticle up converting as well as down converting, which are known for near infrared emission. So today, as I said, I will be talking about this nanodiamonds and the defect centers inside them and the gold nanocluster. So gold nanoclusters. So the gold nanoparticle, it shows a size dependent optical property. It shows a surface plasmon band around 520 nanometer due to the coherent oscillation of the conduction band electron induced by the interacting electromagnetic field of the light. So as you can see, as the size of the gold nanoparticle increases, the color of the gold nanoparticle changes. It becomes from red, it becomes purple and blue. So and you can see here also the absorption spectrum shifts to the higher wavelength. However, when it becomes smaller and smaller, the color become brownish, and then when it becomes less than 2 nanometer, then it become transparent kind of yellow color solution. So as you can see, as the size decreases, the absorption band also decreases because it do not have enough density of states necessary to support the plasmon characteristic of the metal band. So when these nanoparticles become very small, like less than 2-3 nanometer, as you can see the HRTM here, so this behaves like a molecule. So these are not called as a nanoparticle anymore, these are called as a nanocluster. So that's why we say gold AUNC, that means gold nanocluster. So these nanocluster behaves like a molecule. Like if you have a gold metal, like it's a metal, so it doesn't have any band gap, so it doesn't give any fluorescence. But when it becomes such a smaller size, only few atoms, like it has only 25 atoms, so then the band gap opens. So opens up, which can gives the fluorescence. So as you can see here, the fluorescence property of this gold nanocluster depend on the number of atom in the cluster. Like if you have a 5 atom, it shows emission maxima around 385. If you have 8, it shows around 455. And if you have 13, it shows emission maxima at 510. If you have 23, it's around 760. And if it is 31, it's around 866. As you can see, if you put this gold nanoparticles under the UV light, you can see the emitting color. Like you can see here, change from blue, green, and ours is more red color. So it's in the NIR region. So how do we prepare this gold nanoparticle? Like, in earlier days, when initially these nanoparticles were discovered in between 2000 and 2010, people used to prepare the large nanoparticle from tetrachloroauric acid and reduce them to AU0, so AU3 to AU0, and here the citric acid acts as a reducing agent as well as surface coating agent. So Here the citric acid form a coordination bond with gold through the carboxylic acid. So these are not covalent bond, but when we want to prepare such a small, ultra small gold nanocluster, these citric acids cannot stabilize such a small gold nanocluster, it aggregates. So we need sulfur ligand, because gold sulfur, as we know, it forms a very strong covalent bond. So it's a similar procedure. We take the tetrachloroauric acid solution and we put our desired sulfur ligand, and we put sodium borohydride as a reducing agent, and with a vigorous stirring it reduces the Au3 to Au0, and we get our gold nanoparticle solution. So here we use lipoic acid to prepare our gold nanoparticle, because here you can see there is a 2 sulfur here, because instead of using single sulfur, these bidentate ligands are more efficient in stabilizing this gold nanocluster. So we use lipoic acid, and then after preparing the gold nanocluster, we adsorb the polyethylene glycol on it. The reason is because if we stabilize the gold nanocluster only with lipoic acid, it is stable only in basic pH, because in basic pH this COOH group becomes COO minus. So this negatively charged gold nanoparticle becomes stabilized by the electrostatic repulsion. But in acidic pH, the COO minus get protonated and it's precipitated. So, but the lipoic acid is when this polyethylene glycol is attached, it not only gives sterical, it stabilizes the gold nanocluster due to steric reason. So, and when we started this work, we observed the the NIR emitting gold nanoclusters has a very poor quantum yield. So we wanted to improve that, and so our approach. Many groups, what they do, they dope some other elements like cadmium or silver, this, and enhance the quantum yield. Our approach was ligand-based approach. So we attached different ligand on this carboxylic group, and we did a study which group is better in improving the quantum yield. Like we attached positively charged ligand, negatively charged, aromatic group, and one of the ligand which give us a very good enhancement in the quantum yield is this triphenyl phosphonium ion. So when we attach this triphenyl phosphonium ion through the EDC coupling, forming a amide bond, we observe the quantum yield increase from 10% to 25%. So after we observed this phenomena, we studied why this increase, what is the reason behind it. So we also studied the fluorescence lifetime. As we can see, the fluorescence lifetime increased from 126 nanosecond to 466 nanosecond after coupling of this triphenyl phosphonium ion. As you can see, this analysis of the fluorescence lifetime, and the amplitude of the slower decaying part increased quite significantly from 9% to 20%, and here the fast decaying part decreased. And here you can see the electron microscopy image of this gold nanocluster. So you can see the scale bar is 5 nanometers. So the clusters are smaller than the 5 nanometer. It's around 1.5 nanometer. So we also characterize these nanoparticles using Proton Nmr and 31P NMR. As you can see here, like after coupling of this triphenyl phosphonium group, you can see the proton peak from the phenyl group, and here we can see that there is a peak in a phosphorus NMR from quaternary phosphonium ion. So we were sure that these triphenyl phosphonium was intact during our reaction, so it didn't broke or something. So after the characterization, we studied why this increase happened. So we did X-ray photoelectron spectroscopy of this nanoparticle. So as we observed here, the binding energy of the gold, it shifted to the higher energy from the X-ray photoelectron spectroscopy. And usually this shift happens when the gold become more positively charged. And we also did some other experiment, like we varied this counter anion, like we put bromide, chloride, iodide, and tetrafluoroborate. And from all this study, we concluded this enhancement of the quantum yield is due to the charge transfer from the gold core to the phosphonium ion. So this could be a very helpful for preparation of high quantum yield gold nanocluster. The next thing we wanted to study, what happens if we attach some heavy atom on the in the ligand sphere. So how does this affect the optical properties? It is known from the literature, which is called a heavy atom effect, that if you attach a heavy atom to a fluorophore, it enhances the rate of transition of electron from singlet to triplet and triplet to the ground state, singlet ground state. So it is expected, due to heavy atom, the fluorescence lifetime will be faster as you attach the heavy atom. So similarly, we prepared gold nanocluster like my previous slides. So, and after preparation of this gold nanocluster, we attached this 4-bromobenzylamine using EDC coupling. And as a control, we also prepared the this benzylamine. So we kept the number of 4-iodobenzylamine as well as benzylamine equal in both the sample. And we observed that after attaching benzylamine, which is 2, there is an increase in fluorescence. And after attaching the iodobenzylamine, it increases further. And also when we did the lifetime study, we observed that after attaching a benzylamine, it decays slower, but after attaching the iodobenzylamine, which is the blue color, it decays even slower. So this is quite an inverse of what is expected from heavy atom effect, and this phenomena is very rarely observed. So this we call it inverse heavy atom effect. And so we also did this lifetime analysis, and as you can see here, the amplitude of the slow decaying part increases, but the fast decaying component decreases. So as we attach the iodo functionalized ligand. And then we also did study the transient absorption to understand the excited state dynamics of this nanocluster. So as you can see here, the transient spectrum, which is positive for both the particles, which indicates the transient absorption is mainly due to excited state absorption. And here in C, you can see that this is a transient absorption proved at 600 nanometer, and you can see the decay of the excited state is much slower for iodo functionalized gold nanocluster than the benzylamine functionalized nanocluster. So both the nanocluster has a quite similar pattern of excited state dynamics. Like you can see one which is very fast decaying within in picosecond range. Another one is decaying at another component which is decaying in nanosecond or microsecond range. So you can see upon attachment of these iodo ligand, this nanosecond slow component increases more than 6 times. So these very slow decaying triplet state can be very useful for photosensitizer as well as photocatalyst application. So we also tried to explain why it is happening. Like these gold nanocluster usually contain 25 gold atom, and this has a icosahedron in between, which has a 12 atom, and there is a 1 gold atom inside this icosahedron, and there is 6 like there is 12 this AU bond, which we say this is a staple. So usually the short decay, this picosecond decay happens within this icosahedron, and this long decay happens from this staple to the from this staple. So from our this inverse heavy atom effect, we found that gold nanocluster that can be used for photosensitizer as well as photocatalyst application. So then we tried some application. Can this gold nanocluster be used in bioimaging? Like when we incubated the gold nanocluster, we can see it's readily uptaken by the cells, and it doesn't go to nucleus, but we can see in the perinuclear region. We also studied the flow cytometric analysis of this gold nanocluster. So here to detect the gold nanocluster inside the cell using flow cytometer, we needed to do some change in the filter setting. That's what we have shown in this paper. Like usually the all the flow cytometer are developed such a way where the Stokes shift of the dyes are very small. Like if you are exciting at 480, the dyes maybe it will emit at 550 or 580, so it doesn't give much Stokes shift. And the filter setting is usually, when it comes from the company, it's according to that. But for the gold nanocluster, we excite at 405 and the emission maxima is at 760. So there is a say close to 350 nanometer of Stokes shift. So we adjusted the filter setting in such a way, we changed the instrument setting, and then we showed how we can detect the gold nanocluster signal in this in flow cytometric analysis. Like we have shown, like with more incubation time, you can see there's a shift in the signal, and also as we increase the amount of gold nanocluster, we can see there is a shift in the signal. So we can detect quantitatively the gold nanocluster inside the cell using flow cytometer. So now I move to the next topic on the gold nanocluster, is altering the subcellular localization of gold nanocluster by design. So this triphenyl phosphonium ion, so it has a positive charge on the phosphorus, which is delocalized over the 3-phenyl group. So, and this is quite well known to target mitochondria. This triphenyl phosphonium group, it can pass through the cell membrane, and due to this mitochondrial potential, it can specifically target itself and accumulate itself inside the mitochondrial matrix. And this is a widely used strategy to conjugate this triphenyl phosphonium group and target the nanoparticle to the mitochondria. So we prepared 2 types of nanoparticles. Similarly, with lipoic acid, we do from tetrachloroauric acid, we reduced with sodium borohydride, and then we attached the this polyethylene glycol. So if you carefully notice here, the polyethylene glycol structure, one, it is terminated with methyl, and one with terminated with carboxylic acid. So, and the both has a sulfur group. So the sulfur group is attached to the gold. And then what we did, we attached this triphenyl phosphonium group. So in particle 6, you can see the triphenyl phosphonium group are more exposed, and because it is attached to the lipoic acid as well as to the PEG. But in case of particle 5, you can see it's attached to only lipoic acid, so it stays, inside this polyethylene glycol core, so it is not exposed. And then we treated these two types of nanocluster to the cells. As expected, we observed this particle 6, which has an exposed triphenyl phosphonium group. It targets mitochondria. We have done the co-localization experiment. As you can see, it's nicely co-localizing. The red color is the signal from the gold nanocluster, and the green is the signal from the mito tracker. And you can see it's nicely co-localizing. It's giving a yellow color. And then when we put the particle 5 to the cell, surprisingly, we observe it's going to the Golgi apparatus instead of mitochondria. So here is the confocal image. You can see we also did the co-localization experiment with Golgi tracker. This is the zoom in image. So, and we try to find out why this, different behavior. So we studied the cell uptake mechanism, and we observed the particle 5, which goes to Golgi body, is uptaken by clathrin mediated active cell uptake, whereas the particle 6, which goes to mitochondria, it is mainly passive diffusion mechanism uptake of cells. And to study why the 5 is going to the Golgi body, to study the mechanism, we incubate the cell with a molecule. It's called retro-2. So it usually blocks the pathway from early endosome to Golgi body transport. And what we observe when we pre-treat the cell with retro-2, we don't see this accumulation of the nanoparticle in the Golgi body. But when we don't treat them, we see them accumulating in the Golgi body. So we have concluded that this nanoparticle, it's after uptaken by the cell, from early endosome, it goes to Golgi body via this procedure, this retrograde pathway. In summary, I can show what I have shown is this triphenyl phosphonium group enhances the quantum yield of the gold nanocluster due to metal to ligand charge transfer. The presence of heavy atom iodine in the ligand sphere of the AUNC increases the excited state lifetime by 6 times. And I have shown you how we can tune the subcellular localization of the gold nanocluster inside the cell by altering the chemistry of the surface ligand. So now I move to the next nanoparticle that I'm going to talk about is the color centers in diamond. So as we know, the diamond is quite well known for its shininess as a jewelry material. It has a typical sp3 hybridized carbon, and it has a face center cubic structure with a unit cell distance around 0.357 nanometer. So even though it's used for cutting and grinding, it's not that much exciting for scientists. But once you put some impurities such as nitrogen, silicon, or boron, it becomes fluorescence. It becomes optically active, so which becomes very exciting for the scientists for research. In fact, if you know the Hope's diamond, the blue color diamond, which is very famous, it is due to the boron impurity in that. So when we put the nitrogen impurity, so it gives a fluorescence which is in the near infrared region. So it can be used for bioimaging. Also the silicon vacancy center, it's here you can see the nitrogen with one vacancy, whereas silicon, it's a di-vacancy. So you have a silicon and there is two vacancy, one up and down. And also there are many other, this color centers reported from, say, germanium, tin, but these gives emission in the visible regions, say 600 or 630 nanometer. There are, more than 500 color centers are known, but not all have been studied. So like you can see here TR12, which gives emission in the blue region. So this is formed due to interstitial carbon atom in the diamond lattice. So the H3, which gives emission maximum at 503 nanometer. So H3 is basically if you have a two nitrogen atom and one vacancy in between, that is NVN center, and ST1 center, which is not well known, still under study, like what is the impurity atom, we still don't know. NV0, like this is a neutral charge nitrogen vacancy center. There is also the europium europium vacancy center. The negatively charged nitrogen vacancy center, it shows a ZPL at 637 nanometer. And the negatively charged silicon vacancy center, it shows, as I showed you in the previous slides, the ZPL around 737 nanometer. Also, the chromium gives emission in the near infrared region, as well as nickel silicon vacancy or impurity. It also gives emission around 767 to 775 nanometer. And Ne8, this color center, this has a nickel and it's surrounded by 8 nitrogen atom. So this also gives emission in the near infrared region. So these arrow signs indicate these nanoparticles where their optical properties are connected to the spin properties, so which I will discuss with you in the following slides. So recently this SIV0 has also been characterized, which emits in even farther near infrared at 938 nanometer. So I will talk mainly about nitrogen vacancy center because this has been well researched, and I also have worked and currently working on this NV center. So here, if you look into the structure of NV center, you have a nitrogen impurity and there is beside a vacancy. So nitrogen forms 3 bonds, that means it has a 1 lone pair here, and from the beside the vacancy there are 3 carbon which forms 3 bond, and each of them has a 1 unpaired electron. So so this NV center, the neutral charge NV center, it's contain 5 electron, whereas for negatively charged NV center, it contains 6 electron. So 1 electron is sitting in this vacancy. So as you can see in the optical spectra, these are 0 phonon lines of the NV0 and NV minus at 637 nanometer, and these broad spectra is observed due to phononic broadening. So these NIR fluorescence makes this fluorescent nanodiamond. So the fluorescent nanodiamond, I mean, is which nanodiamond which contain NV center. So it's very attractive for bioimaging application because nanodiamond is non-toxic, and also these, these fluorescents are highly stable because the NV centers is deeply embedded inside the diamond. So it can be used for super resolution imaging like STED microscopy, because in this STED microscopy, people use, milliwatt of laser power, which can easily destroy any, fluorescent dyes. So these are very useful for super resolution imaging. As you can see here, they can improve the resolution from 237 nanometer of confocal microscopy to STED microscopy, 39 nanometer beating the diffraction limit barrier. And in this, this NV minus center at 637 nanometer, they emit a single photon per excitation, and it has a very long coherence time of 2 milliseconds at room temperature. So these can behave as an independent quantum system. So this can be very useful for quantum information processing application. And what sets apart the NV center from all the other nanoparticle is called optically detected magnetic resonance, which is ODMR. So as I told you, the NV center, you can read out the spin property by optical spectra. So this is the electronic energy structure diagram of NV center. So it has a A3 ground state and E3 excited state. So the ground state is because it has 2 unpaired electrons, so it is triplet state. So as you can see, the ground state, it has ms0, the magnetic quantum number, and the distance between the ms plus minus 1 is 2.87 gigahertz. So when we put the microwave to the NV center, so the electron gets transferred from ms0 to ms1. So that means there is a almost more than 30% decrease in a fluorescence intensity is observed, which we can see here. Now, if you put a magnetic field, this decrease in fluorescence, it gets splitted because this ms field, ms plus 1 and minus 1 gets splitted. So there will be true transition from ms0 to plus 1 as well as ms0 to minus 1. So as you can see, if you increase the magnetic field, the separation between these two peaks increases. So by this way, you can sense the magnetic field optically, so which is very rarely, or I haven't seen for any other materials. These NV center in nanodiamond can be used for different sensing applications like temperature sensing, sensing of different physiological species like radicals, or magnetic fields sensing of the orientation, the rotational dynamics, as well as the action potential of neuron. So we have recently summarized in one review different application of the NV center in SCS Sensor Journal. If you are interested, you can have a look. So now, like how we create these color centers in diamond. So unlike gold nanocluster, we cannot create this in our lab. So usually we have to visit any DAE center where there is a cyclotron available. So usually we go to with this VECC, Variable Energy Cyclotron Center, which is in Kolkata. So usually we irradiate the diamond target with a energetic nitrogen species, and then it creates a vacancy as well as implant the nitrogen. After implantation, we anneal the diamond at 900 to 1000 degrees centigrade. Then this vacancy center diffuse through the carbon and get merged with the nitrogen, forms NV center. So here you can see one, like last month we did one implantation on a bulk diamond. So this is a high temperature, high pressure diamond, synthetic diamond. So we did we wanted to do very shallow implantation. So we used very low energy like 5 keV, and also we didn't wanted a large amount of NV center. So we wanted we used the fluence of 10 to the power 13 atom per centimeter square, using current in the nanoampere range. So usually we do the SRIM calculation using SRIM software. So how much energy we want, say, if we want in a 10 nanometer depth, the vacancy center, or 20 nanometer. So, and then we use the energy accordingly. So after creation of NV center, like here we use bulk diamond, but we can also use nano diamond. Like on a glass surface, we can prepare a film, and on that we can do the irradiation. After irradiation, the diamond surface contains many different oxygen containing group like carboxylic acid, hydroxylic acid, anhydride, epoxide, or ketone, or lactone. So, and also during this process of ion implantation and the annealing, some of the surface diamond, it gets converted to graphene or amorphous carbon. So these are quite harmful for the fluorescence of the NV center, because the amorphous carbon, it quenches the fluorescence of the NV center. So we do oxidation, we treatment using a very harsh mixture of 1:1:1 mixture of HCl, H2SO4 and perchloric acid. So after the treatment, most of the group gets oxidized and form a carboxylic acid. And these, one might think it's now very easy to functionalize this fluorescent nanodiamond using this amide bond formation or ester bond formation. Unfortunately, when we try to couple any ligand, it get immediately precipitates. Unlike other nanoparticle, once it precipitates, the precipitation is so hard that normal sonication, it doesn't disintegrate anymore. So you need ball milling and etc. So we wanted to functionalize the nanoparticle. To how to do that? To do that, we observed that this carboxylated nanodiamond, it's absorb the the amino acid proteins quite significantly. So we use that strategy, like we absorb polypeptide based polymer onto the surface of the nanodiamond. At pH 8.5, the carboxylic acid is negatively charged and the amine group is protonated, positively charged. So it absorbs this polypeptide polymer using due to this electrostatic interaction. And you can see after coating the FND fluorescent nanodiamond with this polymer, it remains in the solution. It don't precipitate even in 1 molar sodium chloride, like which has a very high ionic strength. So you can see from the TEM image, every single nanodiamond you can see here. So we also showed in this paper, like we conjugated the doxorubicin drug in that, and we also showed the delivery of the doxorubicin drug inside the cell using FND. And there are many other functionalization in last few years that has been developed, like you can reduce the carboxylic acid to hydroxyl group using lithium aluminum hydride. You can prepare amine functionalized, like the biotin, streptavidins functionalized nanoparticle. Also, we can attach some bioorthogonal ligand like tetragen, which can be used, or triple bond, which can be used for click chemistry reaction. And another way to stabilize the nanoparticle, grow a polymer shell on it. So the polyglycerol and HPMA also have been described in the literature, but these polymer shell are quite helpful, not only stabilizing the nanoparticle, but it also prevents the non-specific adsorption of the protein on the surface of the diamond. So and the next thing that we, when we were working, like we wanted to study what happens if we bring a spin, electronic spin near to this NV center. What happens to the T1, T2 relaxation time? Like, so what we did, we adsorbed ferritin protein onto the surface of the diamond. Like ferritin is an iron storage protein. It has 24 fragments and it can store like up to 4,500 iron atom inside it. So we adsorbed the ferritin onto the surface of the fluorescent nanodiamond using the similar procedure. At boric acid buffer pH 8.5, it is adsorbed by electrostatic interaction between the COO minus of FND and NH3 plus of the ferritin. So as you can see here in the high resolution TEM, we can clearly see the ferritin adsorbed onto it. So from the three-dimensional TEM, we observe roughly 5 to 10 ferritin was adsorbed onto the each nanodiamond, and then we study the the relaxation of the spin of the FND NV center. So we observed upon attaching the ferritin, the T1, T2 time decreases significantly, like the spin lattice relaxation time, T1 time decreases from 43.6 microsecond to 23.2 microsecond, whereas the T1, T2 relaxation, the T1, T2, the spin relaxation time decreases from 3.4 microsecond to 0.62 microsecond. This is mainly due to the magnetic noise created by the iron oxide, the iron particle. So in summary, I have shown you the creation of nanoparticle by ion implantation and how the surface functionalization was done, and the detection of few metalloprotein ferritin using FND. So as I promised, before I finish, I will show you 1 or 2 slides of my institute. So we are a autonomous institute of UGC, which aims to facilitate the university utilization of our in-house facility as well as mega science facilities at DAE centers. So our head office is at Indore, and we have 3 centers. One at Indore, Mumbai, which is inside the BRC, and one at ours is Kolkata, and we have a Kalpakkam node at IGCAR. So there are different facilities in my lab, like you can see here, Raman, Globebox, DLS, FTIR, ITC, TCS-PC, and also the UVBs and gamma ray chamber. So you can use all this facility in my lab and in other lab also for free. So, and how you can use it? To use that, you have to go to our website. It's called csr.res.in. Then you go to user portal. And in user portal, once you register yourself, and you can look into the facility which you need for your research, and you can apply for online for this facility, and we will provide you this service measurement for free. So this so you can, if any one want to use, you can contact me. This is my email, and students are encouraged to apply for intensive MSc thesis, and from next year beginning I will be taking PhD students, so any interested candidate can apply. Thank you.
Thank you, Dr. Pramanik, for this wonderful lecture, actually, and special thanks for these last few slides. I hope this will be beneficial for the researchers and the faculties also. So I hope you will be now happy for answering the questions. So participants, you are requested to put your questions in the Q&A, and you may also put your questions in the chat box. And so the panelists, you can just unmute yourself and ask your questions. Okay. Okay, there is.
I am
okay.
Dr. Pramanik. Initially, it was found that functionalization of nanoparticles, generally it was done by sulfur-containing groups or phosphorus-containing groups.
Yes, yes.
Is there any reason? We also did. But nowadays, I think it is changing.
Yeah, it's depend on the which nanoparticle we are using. Like for gold, the gold sulfur is so it's the soft interaction, the covalent bond. But it depends on the which metal you are using. For so it changes. So gold, yeah.
It depends on the metal.
Yes. So it
also,
depends on the chemistry. That's why we are in business. Like we chemist, we all are chemists, I guess. So we can tune the the binding and all this. It depends on the metal.
and when some atom is doped there, quantum field increases.
Yes, there are reports for doping. Like I have seen one literature where they doped cadmium into the gold nanocluster and the quantum field increased.
It is due to charge transfer mechanism.
Yes. I don't remember exactly what mechanism they used, but I have to check it.
Another point, due to heavy atom incorporation, fluorescence quantum field enhances. But generally due to heavy atom effect, due to spin orbit interaction, intersystem crossing occurs.
Yes.
And then fluorescence quantum field decreases.
Yes.
But here,
you are right.
But here it is reversed.
Yes. That's why we termed it as an inverse heavy atom effect. So it is, as you said, it is expected due to enhanced spin orbit coupling. There will be more transfer of electron from singlet to triplet and triplet to singlet. And so from triplet, there will be more loss of photon due to non-radiative decay. So it is expected the fluorescence will decay. But in our case, we observe the opposite. So this is called inverse heavy atom. But this is very rarely observed, I can say.
That means I think their delayed fluorescence mechanism occurs. I think because in the populations in the triplet states, they gathered energy and again revert back to the singlet.
Yeah, that can be one possibility. Yes.
And how energetic elemental species generated to incorporate within the diamond?
So as I showed in VCC, we right now have a very low energy beam, like KeV, like 5 or 10 keV, and there is also some MeV range, energy range. So which as I showed you in the slide, can you see my slide?
Yes, I have seen.
Yeah, so in that slide, like so if we do the stream calculation, so if I have to go say few nanometers, say 5 or 10 nanometer, we will use in keV range. But if I have to usually we don't use MEV because it will be too much. So yeah, it depends on the how much depth you want to go.
Okay, actually I am thankful to UGC DAE because we did much work at Calcutta Center, and
please feel free.
European nanoparticles we have generated there by radiation.
Okay, that's great to know. Probably before my joining. So,
yes, before your joining with your coordinator now.
Okay, Dr. Abhijit Saha?
Yes.
So he's our center director now.
I know. And he also knows me.
Okay. I will tell him about it. Okay, so there are several questions I can see. Like, is there any possibility for using other molecule instead of nitrogen in an? Yes, definitely it's possible. Like we can use silicon, then it will be called SiV center, germanium and tin. So there are many molecules it's possible. So usually if you go to IOP Bhubaneswar, there these beams are available. In fact, we are planning somewhere in January to do some other implantation, other element than nitrogen. So hopefully we will do the study on that. And Professor Amalendu Niyogi asked about, can you explain inverse heavy
He is
atom?
student actually, first year student. So.
Okay, okay. Let me share the slides. Then it would be better. So Amalendu, can you say hi so I can listen to you also?
Actually, you can't listen to him, but then I think Mr. Prem Kumar has to unmute him.
Okay,
Yes.
okay, no problem, no problem. So as you can see from here, like this is a Jablonski diagram. Like as Professor also mentioned, like if you put a heavy atom, there will be, due to the enhanced spin orbit coupling, the transition from singlet to triplet and triplet to singlet ground state, it will be faster. And because the electron is getting transferred from singlet to triplet, and in triplet state there will be much loss of photons. So usually in general it is expected that there will be a decrease in a fluorescence and the lifetime will be much more faster. So this is called heavy atom effect. But in our case, we observe the opposite. Like you can see here in the blue color, the iodo functionalized gold nanocluster, it has a highest fluorescence, and also the lifetime, it is decaying slower. Like the blue color one is the iodo functionalized gold nanocluster. So it is the inverse of what is expected from heavy atom effect. So that's why we term this as a inverse heavy atom effect. I hope I managed to explain you.
Yes, sir. Thank you.
Okay, thank you. So let me check if there is, where is the chat? Yeah, but I cannot see anymore the question.
Shayantan from Shayantan Bhanja actually.
Okay, you show 2 ways of making product number 6, but the other carbo where the can attack the product 6. You showed.
Sir, in the slide number, probably slide number 23 or 24.
Yes.
Just can you?
Okay.
If you can share, then maybe it will be easier.
So, so this one.
It is not shared, I guess. The slide is. You have to share it.
Sir, please reshare your slides, sir.
Okay. Okay. Share screen.
Yeah. Now it's okay.
So this one.
Yes, sir. Proton number 6, you can see. So there are other carboxyl group also in the left side. You can see that on the
yes.
that there the ligand phosphorus is not attaching itself. That
actually this is kind of, it doesn't matter. Like this is a kind of cartoon which we did. There are some carboxylic acid group which has been functionalized. Some of them are not. We characterize by the zeta potential that most of the carboxylic group on the PEG has been functionalized. So this you should take it as a cartoon, not as a quantitative that only the not all of them has been functionalized. This is just to explain you what is the difference between these two particles.
okay, sir.
And there is one more question. Do you able to see that?
Yes, can you explain the ODMR and the graph? Okay, sure. Can you see the slide?
Yes, sir, I can see.
Yes, sir, it is visible.
Okay, okay. So ODMR, like this is the very simplified energy level diagram of the NV center. So you can see the ground state A3. You have a MS0 and MS plus minus 1, and this is the excited state. So MS plus minus 1 as well as MS0. So when you excite, usually the diamonds are excited with a green laser of 532 nanometer. So when you excite them, this gets can go to MS0 and MS1, and then it can go through this metastable A1 state, which is a singlet, and then come to the MS0 state. So the transition probability of MS plus minus 1 to A1 is, it has been observed, is higher than MS 0 to A1 state. So due to this transition, you can see there is a spin polarization. That means the more particle will come, more electron will come and accumulate in MS0. So, and then if at the same time, if you put the microwave here, so this microwave 2.87 gigahertz, it will excite to the ground state only, not to the excited state. So which is MS plus minus 1. So that means there will be, it has been observed almost 30% photon which gets excited to this ground state MS plus minus 1. So now you get a 30% less fluorescence intensity. So that's why when you apply this microwave, you see a dip in a fluorescent intensity. And now once you put microwave as well as some magnetic field, so these MS plus minus 1 get splitted, like MS equal to plus 1 and MS equal to minus 1. So then you will observe two transition, MS equal to 0 to MS plus 1 as well as MS equal to 0 to MS minus 1. So then you will see a split in this dip of the intensity. So as we increase the magnetic field, the separation between these two dip increases. So this what we call it as a ODMR.
Okay, sir, thank you.
Okay, welcome.
Dr. Pramanik, I have one question actually regarding these gold nanoclusters. You mentioned that depending on the number of AU atoms present,
yes,
the properties vary a lot.
Yes,
so
yes.
Is there any way to control the numbers or get the targeted number for the AU cluster, or design any kind of ligand so that any specific number of nanocluster can be stabilized?
Yes, actually, people have developed, this gold business is running for last 2 decades. People has developed a
actually,
method.
That's why that golden fullerene, that's AU32 and AU20, these clusters are quite stable because there are some
people have, the scientists have crystallized them. They got a, the single, only AU25. I can show you one crystal structure, just a moment. So
even in this case also, you have a particular number of AU, I guess.
So here there is AU25 and there are 18 sulfur. So people now know how to crystallize a particular with a specific number of AU atom in the crystal. So it's possible.
Okay, and how is the dimer-dimer interaction in this case? Is there any interaction, or they can be stable as clusters?
They are stable as a clusters. So
okay, thank you.
Okay.
I hope there is no more question. Okay, thank you.
Okay.
Thank you so much for this wonderful lecture.
Thank you for the opportunity. And I hope you have learned a lot, especially for the student. And if you want to use any facilities or do some internship or MSc thesis, you are welcome to apply. Okay, thank you.
Thank you. Now we'll move to our next lecture. Okay, Dr. Deepak Shamanto, he is already here. And Dr. Shamanto, can you, am I audible?
Yes, ma'am.
Okay, so first let me introduce Dr. Shamanto. He has completed his MSc from IIT Madras again, and he has done his PhD from IISc in coordination driven self assembly under supervision of Dr. Parthasarathi Mukherjee, and he has completed his postdoc from two cities. One is from Weizmann, Israel, and another from Netherlands. And now he is working as assistant professor at NISER Bhubaneswar. And his research interest includes reaction in confined spaces, metal organic cages, stimuli responsive materials, and dissipative self-assembly, additive and autonomous materials. So now may I request Dr. Shamanto to please go ahead and present your slides. And he will be talking about reversible photochromism in the cavity of a self-assembled coordination cage. Over to Dr. Shamanto, please.
So thank you, Devasri, for your kind introduction, and also thanks Makeout for giving me opportunity to speak here today. So today I'm going to talk about photochromism in confined space. So chemistry in bulk organic solvent in glass vessel is well known for last few decades. So our interest is to design a molecular architecture which is soluble in aqueous media with a hydrophobic cavity and perform chemistry inside a confined nanospace. In today's lecture, I will mainly focus on photochromic properties of different photo switches inside the cavity of a self-assembled coordination cage. So among many of the photo switches, pyrobiron and azabinin has been most widely investigated. Before I show you our results of chemistry of photochromic compounds inside a self-assembled coordination cage, I'll show you how pyrobiron behaves in normal organic solvents. So pyrobiron generally exists in this ring closed form in many organic solvent, and this is the stable form. And when this ring closed form was exposed to UV light, what happens is that it undergoes ring opening reaction, and it forms this myrcene isomer, and the color of the solution assumed to be deep blue because of very high conjugation in this form, and there is no such conjugation in this ring closed form, but therefore solution is colorless. And this ring open form is very unstable. The lifetime of this ring open form is very short. As soon as this UV light was turned off, this ring open form spontaneously convert back into this ring closed form. And we can easily distinguish between these two isomers using UVBs and NMR spectroscopy. Here you can see the Uvb spectra of pyrobiron and myrcene. So in blue, you can see the Uvb spectra of myrcene, which actually absorb at around 550 nanometer region, whereas pyrobiron, this ring closed form, do not absorb beyond 400. And this is the chemistry of pyrobiron that chemists know for last 50 years. Then the question we asked, can we stabilize this transient myrcene form? And what would be the behavior of pyrobiron inside the molecular vessel? So in the previous slide, we have seen that photoisomerization process is associated with large structural changes, and therefore it requires conformational freedom for this structure inside the confined nanospace, and therefore photoisomerization in confined space is a challenging task. So in our lab, we are designing and synthesizing various molecular architecture with water-soluble molecular architecture with a hydrophobic cavity. Here, for example, we have synthesized this palladium-8 molecular swing and palladium-6 molecular boat, introducing this imidazole-based donor instead of traditional pyridyl-based donor. And this cages has hydrophobic cavity, which can actually encapsulate large guest molecules such as C60, and these cages were utilized for proton conductivity study. So in this case, we have synthesized this palladium-6 triply interlocked cage, and this triply interlocked cage undergo dimerization, 2+2 photodimerization in a single crystal to single crystal pression in presence of sunlight or UV light. And what is more interesting is that this dimerized cage can be separated into its monomeric form by just simply heating its aqueous solution at 90 degrees centigrade. So in this case, we have synthesized a palladium-6 hexanuclear barrel, which was used to catalyze Binswanger reaction and luminescent condensation reaction in an aqueous media. So to investigate this photoswitching property of different photoswitches inside a self-assembled condensation cage, we have prepared this water-soluble molecular architecture with a hydrophobic cavity where we can encapsulate this water-insoluble photoswitches. So it's a very nice cage that one can make by simply mixing these 10 components. This is palladium-based acceptor, and this is triimidazole-based donor, which occupies the wall of the cage, and the corner of the cage are occupied by this acceptor. So this cage not only can be made from these very simple components in a quantitative fashion, the big advantage is that this cage is highly soluble in water because it has 12+ positive charge. And as you can see here, because of the connectivity of this cage, and there's a free rotation between this phenyl and imidazole CN bond, there are various possible conformation of this ring and consequently nitrogen binding site. And because of these two reasons, this cage is now much more flexible, adaptive, and can show breathing motion, shown here by these blue arrows. And because of this breathing motion, now this cage can encapsulate varieties of different guest molecule, including molecule that has many different conformations such as photoswitches. So here are the few photoswitches that we have worked with that can be encapsulated inside the cavity of this cage. So let me just say, we began this project with a series of unexpected results. So this is one of the photoswitch that we have worked with. This molecule is stable in this ring open form because of intermolecular protonation. When we added a yellow solution of this cage into a colorless solution of this cage, we observed the emergence of a deep blue color, which we didn't understand at the beginning. So here's the absorption spectrum. It absorbed at 590 nanometer. So this is because when this switch goes inside the cage, it releases one proton into the solution and gets stabilized inside the cage in this deuterionic form through the pi-pi stacking interaction with the aromatic wall of the cage. When this version of the switch, this ring closed version of the switch was again added to the aqueous solution of this cage, again we observed the emergence of deep blue color. This is because in both the cases, the cage has stabilized this ring open planar mirror form of the switch, which are otherwise unstable. And what is also interesting is that we can convert from this ring closed form of the switch to this ring open form without exposure of any UV light. However, when we added this version of the switch containing a nitro group at this para position, did not result in any color change or any encapsulation. And in a minute, I'll tell you why this is so surprising. So as I have already discussed, this pyroviron switch was developed 50 years ago, and this ring closed form is the colorless form, and this is the stable form. When it's exposed to UV light, it undergoes ring opening reaction, and this ring open mirror form is very unstable. The lifetime of this form is very short. As soon as UV light was turned off, this molecule spontaneously convert back into this ring closed form. Now, I want to bring your attention to this nitro group here. This nitro group normally require to stabilize this molecule in this ring open form because it stabilizes this O minus. And without this nitro group, this molecule is so unstable that it spontaneously convert back into this ring closed form, and in fact, it never be observed. So please note that in the case of molecule that does have a nitro group, in that case, no ring opening reaction is taking place or no encapsulation. Whereas in the case of molecule that doesn't have any nitro group, to stabilize this O minus, in that case, ring opening reaction take place spontaneously, even without exposure to UV light. And to better understand this nature of this process, we have investigated this encapsulation process by Proton Nmr spectra, where we gradually increase the amount of spiropyran into the D2 solution of these cages. Here, these two peaks are due to the cage, and the peak at 0.0 corresponds to the methyl group of this spiropyran. So as we increase the amount of spiropyran into the D2 solution of this cage, you can see here the peak at 0.0 start to increase and become saturated when we have added one equivalent of spiropyran. When we increase the amount of spiropyran, but this peak stop increasing, meaning that cage is saturated with spiropyran, which suggests that cage can only bind with only one equivalent of spiropyran. And it was finally confirmed by single crystal X-ray diffraction study. So what you see here is the single crystal, the cage containing precisely one molecule of spiropyran, and as expected, the spiropyran present inside the cage in this ring open form. And you can also see here the cage has been elongated in the vertical direction, and this is in order to accommodate the guest inside. And you can also see here the cage containing the guest containing a nitro group that we would think would stabilize this ring open form of the switch, but this nitro group will actually collide this part of this cage, and therefore it doesn't take enough and remain in the solution in this ring closed form. So we believe this conformational flexibility of the cage is actually essential for encapsulation of not only spiropyran but varieties of different other guest molecules. So we are interested in the question whether spiropyran retain its photosynthetic property, and indeed we found upon exposure to blue light, the solution turned colorless, meaning that the spiropyran undergo ring closing reaction. So here the absorption spectrum in blue, you can see the absorption spectrum of this ring open mirror form. It actually absorbed at 580 nanometer. However, when we shine the solution at 580 with 580 nanometer of light, we don't see any ring closing reaction. This is because the cage has stabilized this ring open form of the switch. However, this MC form is in equilibrium with this MCH form, which is present very minute amount in this solution. So here's the absorption spectrum of this MCH form in yellow. It actually absorbed at 460 nanometer of light. So therefore, when we shine 460 nanometer of light in this solution, this MCH form, which is present very minute amount in this solution and also is in equilibrium with this MC form, this MCH form actually convert into this ring closed form, and it drives the reaction towards forward direction. And the reaction was reversible as the blue light was turned off. This ring closed form spontaneously convert back into this ring open form. And we have investigated by Uvb spectra. Here you can see with the increase of time, these peaks start to increase and become saturated when we have after 2 hours, meaning that the cage contains actually open form of the switch. So it's a very interesting system. It's based on water, and more importantly, we can make the color disappear by shining blue light, and just upon standing, color reappears. It not only works with the spiropyran, varieties of different other guest molecule as well. So this is adjuvant, this is unsaturated adjuvant. This is one of the best known photoswitchable molecule, and there are a lot of research going on with this unsaturated adjuvant. So when this transform of this unsaturated adjuvant was exposed to UV light, it transform into the cis isomer, and the back isomerization reaction takes place with the blue light irradiation. The same case that we have shown you earlier can encapsulate only one molecule of pyropyran. Now the same cage can encapsulate 2 molecules of adjuvant. So here's the crystal structure of the cage containing 2 molecules of trans-adjuvant, and the adjuvant remain photoresponsive inside the cavity of the cage. So here's the Uvb spectra in black. This is the initial solution of the cage containing 2 molecules of trans-adjuvant, and after shining UV light, we obtain this violet spectra where the peaks due to the trans-adjuvant disappear, which again reappears after shining blue light. And we can go back and forth by shining blue light and UV light. So what I like about this very much is that unsubstituted adjuvant, which has very low solubility in water, which is almost completely insoluble in water, yet in presence of the cage, which has very high solubility in water, we can achieve very high concentrated solution of this adjuvant, and this adjuvant remain photoresponsive inside the cavity of this cage. So it not only works with unsubstituted adjuvant, but varieties of different adjuvant as well. So this is a tetrafluoride adjuvant. So its size is also very small. It is comparable to unsubstituted adjuvant, but because of the presence of 4 fluorine atom at the ortho position, makes the molecule red shifted, now responsive to visible light, specifically green light. So we can achieve trans to cis isomerization using green light, and the back isomerization reaction takes place with blue light irradiation. So here the crystal structure of the cage containing 2 molecules of trans tetrafluoride adjuvant, and again the adjuvant remain photoresponsive inside the cavity of the cage. So in black, this is the initial solution of the cage containing 2 molecules of tetrafluoride adjuvant, and after shining green light, we obtain this green spectrum, and then after shining blue light, we obtain this blue spectrum where the peaks due to the trans adjuvant reappears. So, and the reaction remains reversible inside the cavity of the cage. But there's a difference in isomerization between this unsaturated adjuvant and tetrafluoride adjuvant. We unexpectedly observe precipitation reaction. This is initial solution of the cage containing 2 molecules of trans adjuvant, and after shining green light, we obtain this green spectrum. But what you do not see so well in this picture is that we obtain lot of precipitate. So something else is happening. So when we shine blue light into the solution, the initial solution was regenerated and the precipitate disappear. We analyze the precipitate with X-ray diffraction, and it turns out to be pure cis adjuvant. So apparently, we do not only achieve isomerization from trans to cis upon shining green light, but also precipitation, meaning that the cage doesn't able to accommodate 2 molecules of cis adjuvant within its cavity. So one of the guests has to leave. When we shine blue light into the solution, it re-enters the cage, as it isomerizes back to the trans isomer. Then we decided to take advantage of this and perform the same process in the gel. What you see here is the thin piece of agar gel, which was soaked with our complex containing 2 molecules of trans tetrafluoroazibenene, which was irradiated with a green light different amount of times. 0 second of green light here, 10 second here, 20 second here, 30 second here. As you can see, longer we irradiate, more precipitate appears, and precipitate appearing causes the gel from being transparent to opaque. So we can induce opacity of the gel by irradiating with green light. And then we decided to try to write image on the gel. We thought that by exposing the gel with green light through a mask, we can induce precipitation locally, and the precipitation will cause light scattering, as a result contrast. So we can create a high contrast image by exposing the gel with green light through a mask. And this is how the system works. We took the gel and put a mask on the top of it. Then we shine green light through the mask. And this is the actual image that we have created after shining green light through the mask. The image that we have created will not disappear spontaneously, because unlike the closed form of spiropyran, this cis-azibenene is quite stable. So we have to irradiate with the blue light to erase this image. So this is a truly bistable system. So when we irradiate with the blue light into this solution, so the cis-azibenene that is precipitated will transform into the trans-azibenene, and it will re-enter the cage, and clean solution will be regenerated, and the precipitate will disappear, and you can erase the image. And then you can take another mask and create a new image. So we can create a multiple image by irradiating the gel with green light and blue light. So to quickly summarize this part, we have stabilized this ring open myrocyanine form of the switch, which are otherwise unstable through the piper stacking interaction with the aromatic wall of the cage, and which is also it is also interesting that we can convert from this ring closed form to this ring open form without exposure to UV light in presence of the cage. And we have investigated the photo switching of spiropyran and azibenene, actually many different spiropyran and azibenene inside the cavity of the cage. These photo switches either isomerizes in the cavity of the cage, or in case of tetrafluoroazibenene, isomerization accompanied with the expulsion of one equivalent of gas from the cage. And in both the cases, these isomerizations are reversible. So in the second part of my talk, I'm going to talk about simple supramolecular interaction between very, very simple molecules, not three molecules in solution, but molecule residing on nanoparticle surfaces. So as you may know, in our group, we work quite a lot with nanoparticles. So we specialized in the synthesis of nanoparticles forming varieties of different shape and sizes. So here, for example, you can see different shape nanoparticles of iron oxide. They are all made of iron oxide, but they have different shape. But what you do not see is monolayer of organic molecules that resides on the surfaces of these nanoparticles. So in order to keep these particles stable, they have to be protected with single layer of organic molecules. And these stabilizers are typically long actin chain molecules, and we can function. So there's a possibility that we can functionalize our nanoparticles with functional group of our interest for supramolecular interaction, because we can place different group and at the other end of this molecule. So we are particularly interested in assembling nanoparticles in a reversible fashion. We often work with metallic nanoparticles because it is often the case that these properties of these metallic nanoparticles depends whether the particles are free or aggregated. So we can switch between this property 1 and property 2 depending on whether the particles are free or aggregated. And let's imagine this property 2 that we desire, and using this approach, we should be able to turn on and off this useful property by exposing the system to an external stimulus. Our favorite is of course light, because light can be delivered to a closed system to a particular location and in the form of different colors, which would like to initiate different process in the future. And as opinion, I guess, needs no introduction. This is the transform. When it's exposed to UV light, it isomerizes to the cis form, and the back isomerization reaction takes place with blue light irradiation. This light control conformation changes has been utilized for many switchable host-guest interaction, including mechanically interlocked molecule. Here we'd like to explore a different property change that is associated with the isomerization of azimuthine, that is the change in the polarity of the molecule. The change from hydrophobic nature of trans-azimuthine to this hydrophilic nature of cis-azimuthine. And it is not difficult to imagine condition under which this more polar form will assemble. Its self-assembly structure are dynamic, and this structure persists only under UV light. And we can break the structure by shining blue light. So this is how the system works. So we generally work with nanoparticles. We can place different azimuthine moiety on the surface of these nanoparticles. So if we work with 5.5 nanometer gold nanoparticles, we can put 400 of such azimuthine moiety onto the surface of these nanoparticles. So imagine we are in a hydrophobic solvent such as hexane or methanol. So initially these particles will be soluble in this hydrophobic solvent because they are functionalized with hydrophobic residue, this hydrophobic azimuthine residue. These residues are also hydrophobic, so they are solvated, they are well solvated in this hydrophobic solvent, and therefore these particles will be solvent. But using UV light, we can switch from trans to cis form. As a result, these residues will become a bit hydrophilic and it will lose its solution layer and the particles start to aggregate. These aggregates are dynamic and it only persists when we shine the system with UV light. When the UV light was turned off, or if we shine with visible light, we can break this aggregate. And it turns out it works very well for nanoparticle, many different size and composition. So what we'd like to finish with is to tell you about our alternative approach to this photoswitchable nanoparticle. The approach based on modifying chemically the surfaces of these nanoparticles with the photoswitchable ligand does not always work because of the electronic interaction between the excited state of the photoswitch and metallic nature of the nanoparticle. It is often the case that because of the metallic nature of the nanoparticle, the excited state of the photoswitchable molecules are quenched and back photoisomerization is observed. So we hypothesize we can assemble nanoparticles using light, nanoparticles that are not photoresponsive. And this sounds like an oxymoron, but we hypothesize we could do that by placing these nanoparticles in a photoresponsive media. So in this case, we decided to work with the other photoswitchable molecule that I've already discussed. This is pyropyran. When it exposed to UV light, it isomerizes to this ring open form, and the back isomerization reaction is very fast because of this charge separation. What is also interesting is that we can also open up this ring by adding a little bit of acid into the system, which protonates this oxygen, and now this become thermodynamically stable from. And here's the absorption spectrum. It absorbs at 420 nanometer of light, such that when we expose the system with 420 nanometer of light, it undergoing closing reaction. And what is more interesting is that now this proton is released to the solution. So this molecule often known as photoacid because its solution is not acidic unless we irradiate with light. On the other hand, here we decided to work with nanoparticles that are certainly not photoresponsive. Nanoparticle functionalized with ligand terminated with COH group. And you believe it or not, these particles are insoluble in all solvent, including water and methanol, because of the extended hydrogen bonding interaction between this COH group. However, we can dissolve this particle by adding little bit of acid into the system, which protonates these COH groups and breaks the hydrogen bond such that the particles become soluble, because there are no more hydrogen bond between them, but also they repel each other electrostatically. Then we can use a base which can extract this proton and the particle become assembled again. So we can induce reversible assembly and disassembly of this particle by adding acid and base into the system. But this is not so convenient because each and every cycle will generate waste, and this is not so convenient, and it doesn't work after very few cycles. So here we are basically combining previous two slides. Here we have assembled particles and photoacid. By using light, we can induce a ring closing reaction of this photoacid. As a result, it will release proton into the solution, and this proton will protonate this carboxylate acid group. As a result, there will be and it will break the hydrogen bond between these COH groups, and the particle become disassembled. When the blue light was turned off, this molecule, this ring closed form of this molecule will act as a base, and it will abstract this proton, and the particle become assembled again. So the particles remains in the disassembled form when the system was exposed to UV light. It turns out it works very well for nanoparticle many different sizes, including 5.5 nanometer gold nanoparticles, 2.5 nanometer gold nanoparticles, also 10 nanometer gold nanoparticles. So this is how the system works. This is the ground state of the system. You can see here particles are completely quantitatively assembled. They have precipitated from the solution, as you can see here, and the yellow color of the solution is because of photoswitchable medium. When we shine blue light into the system, this photoswitch undergoing closing reaction, and it's release one proton into the solution, which protonates this particle, and the particle becomes disassembled, and hence the red color. When the blue light was turned off, they quickly reassemble, and they precipitated from the solution as well. It turns out that this system is remarkably reversible, and this is the Dls experiment you can see here, and it was also confirmed by TEM study. This is the ground state of the system. You can see here the particles are completely quantitatively assembled, and after shining blue light, they completely quantitatively disassemble. We can do this not only in solution, but also inside gel. We have prepared robust rigid gel, organogel, and the red color of the gel because the gel was kept near a source of light, which induces the disassembly of this particle. Hence, the gel is red color. And doing this disassembly inside the gel gives us a possibility to expose the system locally. And here we decided to use this mask featuring Cheshire Cat from Alice in Wonderland, appearing, disappearing image. And this is how the system works. We took the gel and put a mask on the top of it, and then we shine blue light through the mask, and the red color appear only in the region where it exposed to blue light, which induces the disassembly of this particle, and hence the red color. And when the blue light was turned off, these particles, these nanoparticles quickly reassemble, and the image that we have created here will slowly disappear, and precisely after 3 minutes you can see here the image was gone. So we can take another mask and create a new image. And at that point we realize that there are several flags which are made of yellow and red. So we take a mask of flag of China, and we have created this image, which again persists for only 3 minutes, and after which it was gone. And then we have created the image of flag of Catalonia, Macedonia, Vietnam, etc. So it's a very interesting system, and we can use it as a reusable information storage media, where each information can be stored only for 3 minutes, and after which it will go, and there is no way you can get it back. And we are very excited that the system works in water now. Previous few slides were devoted to methanol. It's good to have it work in water, because one of our long-term goal is to interface our system with the biological molecules. So I'd like to thank my supervisor, Professor Parthasarathi Mukherjee, and postdoctoral supervisor, Professor Rafal Klan. And I would also like to thank NISER for allowing me to start my group at NISER, and also for their generous funding. And I'd also like to DST ACRB for startup research grant. So thank you.
Thank you, Dr. Shamanta, for this nice talk. And now the session is open for question and answer. And actually, participants, you may put your questions in the chat box or in the Q&A. And panelists, you can just unmute yourself and ask the questions.
Dr. Shamanta?
Yes.
Now, during functionalization of gold nanoparticle,
yes,
by azobenzene,
yes,
you have used a sulfur containing group.
yes,
Which group?
thiol,
What?
thiol.
Oh, thiol. So at first you functionalized with thiol and then interact with azobenzene, or azobenzene?
No. We'll make the ligand first, then we make the ligand with thiol, azobenzene, then we actually gold nanoparticles will prepare with the DDAB and dodecylamine, and then we put the ligand on the surface. Then this thiol ligand will actually remove the amine-based ligand.
Okay.
Because the interaction with gold nanoparticles with thiol will be much more stronger than amine-based.
And that is the reason everybody generally functionalize gold nanoparticles with thiol, because interaction is high.
Yes.
And another point is, due to in your picture shown here, you have used for self-erasing images polyethylene glycol gel.
Yes.
Which polyethylene glycol? It's molecular weight?
Around 4,000.
4,000. Okay. That means,
so I have to check exact. I can send you the exact details. So it's around 4,000. We have used 2 different type of PEG compound and make a gel out of it, and then we have created, because here the systems are in methanol, so we cannot use water-based system.
Because the gel will be like some solid type.
Yes.
Yeah, that is why you high molecular weight. And another point, you have used spiropyran. So when spiropyran, you used light for the conversion.
Yes.
That is its ring is open.
Yes.
And in the cage, it exists as plus and minus. That means it exists as both the charges are there.
Yes.
So in this condition, its stability is there.
Yes, cage is actually stabilizing this planar form through piper stacking interaction. So when you have spiroform, it is not planar. But when you have this planar form, through the aromatic wall, cage is stabilizing this planar form of the
because it is in the Jutter ionic form.
Yes. And cage is also positively charged, because the palladiums are positive. So, and although the cage is positive charge, they have long aromatic wall. So here we have used this tripyridyl aromatic based wall. Because of this piper stacking interaction, we have the crystal structure here. So you can see they have.
Yes, that means aromatic wall is sealed.
Yes, aromatic wall, it will stabilize this ring open form of the switch.
Thank you, very nice work. Good.
And we have another question in the Q&A. Should I read out it for you, or you may see the Q&A part also?
Just 1 second. Okay.
Can you see the question?
Yes. I can see why the molecules with NO2 group shows no ring opening and no encapsulation. So this is because I'll show you here. So as we expected that the molecule with nitro group will be more stable. The ring open form of the switch is more stable, but you can see here in the crystal structure, when we have nitro group at this
Your
para
slide is
position,
frozen,
you cannot see the slide.
We are seeing the slide number 8 here.
Yes, 8.
Okay, yeah. Oh, pointer is missing. That's why maybe we could. Okay, please continue.
So you can. So we would think the switch containing a nitro group would stabilize this ring open form because it stabilizes this O minus. But you can see here, if you have nitro group at this position, it will collide with this part of this cage, and therefore it doesn't get encapsulated inside the cage and remain in the solution in the ring closed form. So
Here, if we increase the cage size, that may possible that.
Yes, definitely. You need to increase the cage size. So we'll be continuing the work later on with the higher cage size.
And is there any kind of interaction that is specifically choosing this molecule? Means if there are other, or it's just the cage size which is just fitting exactly?
No, it has to fit, and also there is pi stacking interaction. With that, it will stabilize the ring open form. Without that interaction, it cannot stabilize the ring open form of the switch.
Okay. And then we think we don't have any other questions. So let us now thank Dr. Samanta for this wonderful talk. And we hope to see you in person maybe when the institute will be opening.
Yes, thank you. Thank you, Devashree, for your kind introduction, and thank you, Makeout, for giving me opportunity to speak here today. So thanks.
Thanks. So with this, we are now closing this session 2, day 2 program, and we will see tomorrow. We'll again meet tomorrow at 10 for the next lecture, session 1, day 3. And Dr. Tapush Ghosh, are you there? Is there any more announcement or anything?
Yeah, nothing. Just would like to thank the speaker again, as you did as well, for taking his time to deliver his talk and his research work with our students. And I hope he has enjoyed delivering the lecture and the interaction session. And in future, we will have the opportunity to listen him more and future works. And for the participants, I think the feedback form that need to be shared for this session. Is it already done? I guess no, not yet. So I think if Prem can...
we should provide it, or we have already provided.
Okay, you have already provided it, no? Okay.
No, sir. In the first session we have given, should we provide it once again?
No, it's okay then. It's okay. It's once a day, it's fine. No problem.
Yes, sir.
It's done, then I think we have nothing more from this session. And we will join as Dr. Manna